Welcome to our first lecture, What is Cancer? In this lecture, we will begin with a basic definition of cancer, look at how cancers are classified, introduce the hallmarks of cancer, and then use breast cancer as an example. We will also connect cancer biology to genomics and big data research, and finish with your first activity.
The human body is made up of trillions of cells. These cells work together to form our tissues and organs. Under normal conditions, cells grow, divide, and die in a very controlled way. But sometimes cells stop following these normal controls. They continue to grow and divide when they should stop. These abnormal cells may eventually form a tumor. Some cancer cells can also spread from their original location to other parts of the body. So the key idea to remember is this: cancer is the uncontrolled growth of abnormal cells. Cancer is not a single disease. In fact, there are many different types of cancer. One way we classify cancers is based on the type of cell or tissue where the cancer begins. As you can see in this figure, the major categories include carcinomas, sarcomas, myelomas, leukemias, lymphomas, and several other types. An important distinction to keep in mind is that we can classify cancer by its tissue of origin, but we can also describe cancer by the organ in which it occurs. On the next slide, we'll look at cancer from that organ-based perspective. Another common way to describe cancer is by the organ where the cancer occurs. As you can see in this figure, cancer can occur in many different organs, including the thyroid, colon and rectum, bladder, uterus, kidney, brain, stomach, lung, and liver. This is probably the way we are most familiar with naming cancers, for example, lung cancer, breast cancer, or liver cancer. However, remember what we discussed on the previous slide. Cancers can also be classified according to the type of cell or tissue where the cancer originated. Next, let's look at how frequently different types of cancer occur. This figure shows how frequently different types of cancer occur in men and women based on statistics from the American Cancer Society. I don't expect you to memorize these numbers. Instead, I want you to look at the overall pattern. You can see that some types of cancer are much more common than others. You can also see that the patterns are different between men and women. These kinds of statistics are important because they help us understand the overall burden of cancer in the population and help guide cancer prevention, screening, and research. Now that we have looked at the different types of cancer and how commonly they occur, let's come back to our basic question. What exactly is cancer?
Now let's come back to our basic question, what is cancer? Cancer is the uncontrolled growth and spread of abnormal cells, and it can arise in essentially any tissue or organ in the body. As we discussed earlier, cancers can be classified into several broad categories, including carcinomas, sarcomas, leukemias, and other types. Carcinomas originate from epithelial tissues, and they account for about 80 to 90% of all cancer cases. But what causes a normal cell to become a cancer cell? At the molecular level, cancer is associated with mutations in our DNA. Some of these mutations can be inherited from our parents, while others are acquired during our lifetime. This brings us to an important question. What changes allow these abnormal cells to behave like cancer cells? We'll look at that next through the hallmarks of cancer.
So, what makes a cancer cell different from a normal cell? Researchers have identified a set of biological capabilities known as the hallmarks of cancer. Here we see 6 major hallmarks. Cancer cells can sustain proliferative signaling, meaning they continue receiving signals to grow and divide. They can evade growth suppressors, essentially ignoring signals that tell them to stop growing. They can resist cell death and achieve replicative immortality, allowing them to continue dividing. They can also induce angiogenesis, which means stimulating the formation of new blood vessels to support tumor growth. And finally, cancer cells can develop the ability to invade surrounding tissues and metastasize to other parts of the body. These hallmarks give us a useful framework for understanding the different changes that allow a normal cell to develop into a cancer cell. And, as we'll see on the next slide, this original list of hallmarks can be further expanded. The hallmark framework can be further expanded as our understanding of cancer biology developed. In addition to the 6 hallmarks we just discussed, several other important characteristics were recognized. Cancer cells can avoid immune destruction, meaning they can escape detection and elimination by our immune system. Tumor-promoting inflammation can create an environment that supports cancer growth. Genome instability and mutation allow cancer cells to accumulate additional genetic changes, and cancer cells can deregulate cellular energetics, changing the way they obtain and use energy to support their rapid growth. The important point here is that cancer is a very complex disease. A tumor usually does not result from a single abnormal pathway. Instead, many biological processes interact with one another as cancer develops and progresses. This complexity is also why cancer research involves so many different areas, which we'll look at next. Because cancer is such a complex disease, cancer research covers many different areas. Some researchers focus on the basic mechanisms of cancer. They ask questions such as, what mutations cause cancer? And how are cancer cells different from normal cells at the molecular and cellular levels? Other researchers study metastasis and invasion, how cancer cells leave the original tumor, spread through the body, and colonize other tissues. Another major area is early detection and diagnosis. Researchers look for biomarkers in blood, urine, or saliva and develop better imaging and pathology methods to detect cancer. Of course, a major area is cancer therapeutics and drug development, including chemotherapy, immunotherapy, hormonal therapy, and radiation therapy. There is also precision oncology, where treatment is selected based on the characteristics of an individual patient's tumor, as well as survivorship, supportive care, clinical trials, and translational research. Throughout this course, we will encounter several of these areas and see how molecular biology and biological data can help us understand cancer. Now let's take one specific cancer, breast cancer, as an example.
Now let's use breast cancer as an example to see how some of these general cancer concepts apply to a specific disease. Breast cancer is relatively common. About 1 in 8 women will develop breast cancer during their lifetime, and the risk increases with age. How do we detect breast cancer? The most commonly used screening method is a mammogram, which is a specialized type of X-ray imaging of the breast. MRI, or magnetic resonance imaging, can also be used in certain situations. If imaging identifies a suspicious area, a biopsy can be performed. A small sample of tissue is removed and examined to determine whether cancer cells are present. Once breast cancer is diagnosed, 2 important questions are, how far has the cancer progressed and what type of breast cancer is it? This brings us to cancer staging, which we'll look at next.
Once breast cancer is diagnosed, one of the important questions is, how far has the cancer progressed? This is what we mean by cancer staging. For breast cancer, we commonly use stages from 0 through 4. Stage 0 refers here to ductal carcinoma in situ, or DCIS. The abnormal cells are still confined within the milk ducts and have not become invasive. In stage 1, the cancer is still relatively small and is generally limited to the breast, although a small number of nearby lymph nodes may be involved. In stage 2, the tumor may be larger, or the cancer may have spread to additional nearby lymph nodes. Stage 3 is considered locally advanced breast cancer. The cancer has spread more extensively to nearby lymph nodes or tissues, such as the chest wall or skin. Finally, stage 4 is metastatic breast cancer. This means that cancer cells have spread from the breast to distant organs such as the bone, liver, lung, or brain. So the important point is that cancer staging tells us the extent to which the cancer has progressed and spread. But stage is not the whole story. Breast cancers can also have different subtypes, which we'll look at next. We just talked about the stage of breast cancer, which tells us how far the cancer has progressed or spread. But breast cancers can also be classified into different subtypes based on their molecular characteristics. One important way to define these subtypes is by looking at specific receptors expressed by the cancer cells. These include the estrogen receptor, or ER, the progesterone receptor, or PR, and a protein called HER2. Different combinations of these markers define different types of breast cancer, and this information is very important because it can help determine which treatments are most likely to work. So, two patients may both be diagnosed with breast cancer, even at a similar stage, but their cancers may be biologically quite different, and therefore they may receive different treatments. This is an important example of what we call precision oncology, using the molecular characteristics of an individual tumor to help guide treatment decisions.
Now, let's shift our attention from cancer biology to cancer data. A major turning point came with the completion of the Human Genome Project around 2003. At the same time, DNA sequencing technologies advanced rapidly, and the cost of sequencing became much lower. These developments made it possible to generate enormous amounts of genomic data from cancer patients. One particularly important project is the Cancer Genome Atlas, or TCGA, which was launched in 2006. TCGA collected and generated large-scale molecular data across many different types of cancer. This fundamentally changed the way we can study cancer. Instead of studying one gene in a small number of samples, researchers can now examine thousands of genes across hundreds or even thousands of patients. This is what we mean by big data in cancer biology. To understand these genomic data, however, we first need some basic knowledge about the human genome. So let's review that next.
To work with cancer genomics, we first need to review a few basic facts about the human genome. Humans have 23 pairs of chromosomes. These chromosomes contain our DNA, and the entire human genome contains roughly 3 billion DNA base pairs. One of the major accomplishments of the Human Genome Project was to sequence the human genome and provide what we call a reference genome. You can think of the reference genome as a kind of map or coordinate system for our DNA. It allows us to ask questions such as, where is a particular gene located? What DNA sequence is found at that location? And how does the DNA from a particular sample differ from the reference? This reference becomes extremely useful when we study cancer because we can compare genomic data from cancer cells with the reference genome and identify changes or mutations. Next, let's look briefly at how the Human Genome Project produced this important resource. Let's look briefly at the Human Genome Project itself. The project began in 1990 and was completed by 2003. At the time, this was an enormous scientific undertaking involving both gene mapping and DNA sequencing. One of its most important outcomes was the establishment of a reference sequence for the human genome. I've included a link on this slide to the Genome Data Viewer. I would like you to actually visit this website and explore the human genome for yourself. As you explore it, I want you to think about two questions. How many genes do humans have? And where are these genes located across our chromosomes? You will see chromosomes 1 through 22, as well as the X and Y chromosomes. You will also see MT, which represents the mitochondrial genome. Take some time to zoom in and look at individual genes. The goal is to become familiar with how genomic information is organized and how we can actually access and explore the human reference genome.
For your first hands-on activity, I'd like you to choose one type of cancer and do some additional research about it. In your write-up, first explain how common this cancer is in the population and how it is typically detected or diagnosed. Then discuss any known risk factors that may increase a person's chance of developing this cancer. I also want you to explain the staging system used for this cancer and describe any important subtypes. Next, summarize the currently available treatments and look into the typical survival or life expectancy information for patients. The goal is not simply to collect a list of facts. I want you to organize the information into a clear, understandable explanation of one cancer type. Once you complete the research, you will publish your write-up on your personal GitHub blog. On the next slide, I'll show you where to find instructions for setting up that blog. Now, how do you create your personal blog on GitHub? I've provided 2 examples on this slide to help you get started. First, visit my personal GitHub blog. There you can find the tutorial that I used to create the site. I've also provided a link to a former student's GitHub blog where you can find another tutorial explaining how to set up a GitHub page. You can follow either of these examples to create your own site. Don't worry if you have never used GitHub before. The purpose of this activity is also to give you some experience using GitHub and creating your own online scientific content. Once your blog is set up, you will use it to publish the cancer research article from your first activity. And that brings us to the end of our first lecture.